Hey class, let's get started with our discussion on the muscular system. Our muscular system is going to be comprised of three different kinds of muscular tissue. And this is reviewed from earlier on when we talked about different tissue types. The three tissue types of our muscular system include skeletal muscle, smooth muscle, and cardiac muscle tissue. Of these tissue types, skeletal muscle is the most common. It's also voluntary. You choose when to contract it. And as its name implies, skeletal muscles are attached to some part of the skeleton. Some kind of bone will be attached to skeletal muscle tissues. Cardiac muscle tissue is the muscle tissue found in the heart, and thank goodness it is involuntary. And the same thing for smooth muscle. Smooth muscle is found in all of the hollow organs and blood vessels of our body. And when we think of smooth muscle, I want you to think of blood vessels, I want you to think of digestive organs, I want you to think of respiratory organs, I want you to think of glands that squeeze secretions out. These are all going to be related to smooth muscle functions, and smooth muscle is involuntary. You do not choose when to contract your smooth muscle tissue. As we look at these three different muscle tissue types, skeletal muscle tissue is going to be multinucleated. Each individual muscle cell has multiple nuclei in it. This is a snapshot of an individual muscle cell. Individual skeletal muscle cells are quite long, up to 10 or 15 centimeters in length, and they also have a high degree of striations. They'll have lots and lots of light pink and dark red lines to them. Smooth muscle tissue, on the other hand, is mononucleated, and it is fusiform in shape. When I say fusiform, I mean that it starts at a tapered end, bulges in the middle, and then goes to another tapered end. And then we have cardiac muscle tissue, cardiomyocytes. Cardiomyocytes are typically going to be mononucleated. Occasionally they're binucleated, and there's also a branching structure that occurs to some but not all cardiomyocytes. When we look at cardiomyocytes or cardiac muscle tissue, there's a very important structure known as the intercalated disc, which represents as a single dark band that separates adjacent muscle cells. And that intercalated disc functions as an electrical synapse. In other words, electrical charges can go directly from one cardiac muscle cell directly into the next cardiac muscle cell. So that when one cardiac muscle cell is electrified, that electric charge will spread to all of the adjacent cells. Some of the functions of the skeletal muscles include supporting the body. They allow us to remain upright. They maintain our posture. Skeletal muscle tissues also allow for movement by attaching to the skeleton, and I should also emphasize that they allow for movement by contracting. So all muscle tissues can contract. They will never be able to push, but they can pull. So when these skeletal muscle tissues allow for movement, we have opposite muscles, they're called antagonistic pairs, that will contract to move a joint. We also are going to maintain a constant body temperature, in large part due to our skeletal muscle tissues. When we get to the molecular mechanisms of contraction, we'll talk about how there's lots of energy used up, and there's a little bit of waste energy or heat generated in this process, and that waste heat maintains our body temperature. Our skeletal muscle tissues allow for movement in cardiovascular and lymphatic vessels. It's known as a skeletal muscle pump that in particular helps move blood through the veins and lymphatic vessels. And then finally, we stabilize joints with our muscles and protect internal organs with our skeletal muscles. Those are relatively self-explanatory. I need to strongly emphasize this. All muscles are attached to a bone with a tendon. It's a muscle-to-bone connection. Now, there are a couple exceptions, but generally in this course, we're just going to say tendons connect muscles to bone, and we are not going to focus on the exceptions. All skeletal muscles are going to have two ends to them, an end that's attached to a stationary bone and an end that's attached to a moving bone. The end that's attached to the stationary bone is the origin, and the end that's attached to the moving bone is called the insertion. A good rule of thumb is that the origin, generally speaking, is closer to the heart, and the insertion, generally speaking, is farther away from the heart. The major exception to this is going to be muscles of the face. That rule of thumb doesn't apply to, but for most of the other muscles of the body, generally speaking, the origin is the end of the muscle closer to the heart, and the insertion is the end of the muscle farther away from the heart. When we move our muscles, we can contract them. So if I am flexing my elbow right here, I'm going to contract the biceps brachii, and when I extend the elbow, I contract the triceps brachii. And because they work opposite of each other, these are going to be called an antagonistic pair or antagonistic muscles. We also have synergistic muscles that can work in conjunction with each other. Not shown here are some other muscles of the upper arm. There is the coracobrachialis and then the brachialis and the brachioradialis. Those are other muscles that help the elbow to contract. The primary muscle that helps the elbow to contract is the biceps brachii, but those other muscles that I mentioned all work together to help that joint to contract. As we're naming our skeletal muscles, we name them based off a variety of different factors: size, shape, attachment point, so on and so forth. So let's go over some specific examples. If we look at the big muscles of the body usually are going to have a term called maximus in their name. For example, the gluteus maximus is the largest buttocks muscle. Another term that's related to the size is going to be magnus, medius, and minimus, depending on the size of the muscle. We also can name a muscle after the shape. Usually we'll have sheet-like or triangular muscles. The Greek letter delta looks like a triangle. So if we look at the muscle of the upper arm, the shoulder, the deltoid, which is triangular in shape, we named it after the Greek letter that's also shaped like a triangle. Many times we'll name the muscle based off of the bone it's located over. A good example would be the temporalis, the occipitalis, and the frontalis. Those three bones are going to be over the occipital bone, the temporal bone, and the frontal bone of the skull. We can also name bones after the direction of the muscle fibers that are present in that bone. For example, if we look at the rectus abdominis, all of those muscle fibers in the rectus abdominis are all longitudinal and parallel with each other. And because they're parallel or straight with each other, we use the Greek root for straight, which is rectus, and we call it the rectus abdominis. It's rectus for straight, abdominis because it's over the abdominal cavity. So the rectus abdominis is a twofer. It's named for the direction of the muscle fibers, and it's also named for the location of the muscles. We can also name our muscles for the attachment points. Classic examples include the biceps brachii and triceps brachii. Oh, excuse me, that was the number of attachments. I'm stealing my thunder here. So if we name the muscles for the number of attachments, we can look at the biceps brachii and triceps brachii. Biceps brachii, bi meaning 2, will have 2 attachment points. Triceps brachii has 3 attachment points. We also can name them based off of the locations they are attached to. The brachioradialis is a muscle that starts in the brachial or upper arm region and is connected directly to the radius. Another classic example of attachment muscles include the sternocleidomastoid. It attaches to the sternum, goes over the clavicle, and attaches to the mastoid process. Sternocleidomastoid. It's one of my favorite muscles. Anyways, I digress. We can also name our muscles after the action that they perform. For example, the extensor digitorum is going to extend the digits. We also have the flexor digitorum as well. And another one, the extensor digitorum longus, it will extend the toes, and it's really long in shape, and we had to differentiate that one from the extensor digitorum, which extends the fingers. Both fingers and toes are called digits, FYI. Here are a lot of muscles of the human body. For lab purposes, we are going to be memorizing all of these muscles in lab. But since this is a lecture recording, I'm not going to spend a lot of time reading this figure out loud for you. If you want to spend some more time going over it, go ahead and pause the video, and then you can go over these muscles in more detail. Concept check. So, class. Which of the following is not a way that muscles are named? Are they named? We have size, shape, location, and tissue type. So go ahead and pause the video and get me an answer. You can rewind it, check your notes, check your textbook, or you can phone a friend. Go ahead and get me an answer right now. 5, 4, 3, 2, 1. Muscles are not named for their tissue type. Muscles are named for size, shape, and location, but they are not directly named for tissue type. Moving on, we have muscle fibers, aka muscle cells. And this is something that caused me a lot of confusion, a lot of undue stress when I was a student. To me, it's more intuitive just to call it a muscle cell, but because it's long and stringy, it's also intuitive to call it a muscle fiber. I understand why both names are used. I just prefer calling it a cell. So when we refer to a muscle fiber, know that I mean a muscle cell. And because we have to have special names for lots of things, we are going to have some special names for parts of the muscle cell. For example, we can't call it a cell membrane or plasma membrane. We need to call it a sarcolemma. Sarco is a prefix meaning muscle. And we can't just call it the cytoplasm of the muscle cell. We need to call it the sarcoplasm. And we can't call it an endoplasmic reticulum that releases calcium. We need to call it a sarcoplasmic reticulum because it's unique to muscle cells. As an aside, if you look at the word sarcastic, it has the same sarco word in it. Sarcastic literally means in the Greek to tear flesh from the bone. So think about the next time you're trying to be sarcastic to somebody. It's not the nicest thing to do. Anyways, I digress. As we look at our muscle fibers, there's some terminology that stands for the whole muscle itself. So first, we were talking about terms that applied here on this screen to the individual muscle cells. These terms apply to the whole muscle themselves. We'll switch from the sarco prefix to a myo prefix, and myo also means muscle. The general arrangement of muscles, whole muscles and muscle cells is going to be a bundle of bundles arrangement. So first we start with the whole muscle, and the whole muscle is going to have bundles inside. Is going to be a bundle of bundles, and the bundle inside is called a fascicle. So I'm just going to skip to this figure and talk about it because it's easier if I talk with the actual photograph. So here's the whole muscle. That whole muscle contains individual bundles. Each of those individual bundles is called a fascicle. And if you zoom in on one individual fascicle, that individual fascicle is going to contain bundles, or is going to contain many individual muscle cells, aka muscle fibers. And if we look inside of an individual muscle fiber, it contains a myofibril. A myofibril is inside of the muscle fiber, inside of the muscle cell. And here's the myofibril again. As I zoom in on that myofibril, it's going to be made of thick and thin myofilaments. And as I zoom in on those myofilaments, we find that the myofibril is made of repeating segments known as sarcomeres. So we have a sarcomere that starts right here at a Z-disc, and it will continue to the next Z-disc, and then the sarcomere continues to the third Z-disc, which is not shown, to be about right over here. This structure, this sarcomere structure, with overlapping thick and thin filaments, forms the banding pattern that you see in skeletal muscle tissues. Within that sarcomere, the two myofilaments, the thick and thin filaments, have two primary proteins associated with them. The primary protein associated with the thick filament is called myosin. And each myosin molecule is aptly shaped like a golf club. I think it's a great analogy, where the head of the golf club is going to be the head of the myosin molecule. The thin filament is composed primarily of a protein known as actin. And myosin and actin, these two primary proteins, are going to slide over each other when we contract the sarcomere and therefore contract the muscle. So here, if we look at a relaxed sarcomere, there is not a lot of overlap between the myosin filament and the actin filaments. And when we contract the sarcomere, those filaments overlap by an increasing degree. There's more overlap between them, and that causes the sarcomere to get shorter, and then the muscle to get shorter as a whole. When we start to contract a muscle, we need to take an electrical signal from the motor neuron and then send that electrical signal to the muscle fiber, to the muscle cell directly. The neurotransmitter that's used to do that will take that information from the motor neuron to the muscle cell, is called acetylcholine. Acetylcholine is notable in that it's the most common neurotransmitter in the body. It's also the first neurotransmitter that was discovered. And when we have that acetylcholine, that chemical released from the motor neuron, and when acetylcholine touches the muscle fiber, that acetylcholine will cause the muscle fiber to have an electrical signal travel across it and then release calcium from the SR, the sarcoplasmic reticulum, within the muscle fiber. So here we can see a motor neuron coming in contact, one motor neuron branching to touch several muscle fibers. The axon terminal or axon bulb will come in contact with the muscle fiber, and then there'll be synaptic vesicles filled with acetylcholine. That acetylcholine will be exositized to the neuromuscular junction and then travels across the gap, across the synapse, and binds to acetylcholine receptors. And by the way, the abbreviation for acetylcholine is capital A, capital C, lowercase h. And as that acetylcholine binds to the acetylcholine receptor, that initiates the electrical signal that moves across the skeletal muscle fiber, initiating contraction. After that calcium has been released from the sarcoplasmic reticulum, it binds to a protein that we haven't talked about yet. This protein is within the sarcomere, particularly on the thin filaments. This protein is called troponin, and when troponin binds to calcium, or calcium binds to troponin, that causes troponin to change shape and pull an attached protein called tropomyosin out of the way so that actin and myosin filaments can come in direct contact with each other. When the myosin heads come in direct contact with the actin filaments, that's going to start a cycle known as the cross-bridge cycle. ATP will bind to the myosin heads, and it's used, the energy in that ATP is used to cause the myosin heads to bend back and forth, pulling the actin filaments towards the center of the sarcomere. And once those actin filaments are being pulled towards the center of the sarcomere, that's what initiates contraction. So here is a thin filament, mostly actin. Calcium can bind to troponin in blue, and when calcium binds to troponin in blue, troponin changes shape and pulls tropomyosin out of the way to expose myosin binding sites. Myosin will then bind to the binding sites on actin and then start bending back and forth to slide the actin past its surface, and ATP is used up in that process. It's very similar to someone pulling on a rope during tug-of-war, where actin represents, or the rope represents the actin filament, and the individual hands of the team on the tug-of-war team pulling on the rope represent individual myosin heads pulling on the actin filament. We need ATP for several different things during muscle contraction and during muscle relaxation. We need ATP to detach the myosin head from the actin. Step number one. We also need ATP to recharge or re-energize the myosin heads so that they can continue the contraction process. Now, let's say, though, somebody dies. If they die, there'll be a little bit of ATP produced for a little bit of time. However, eventually, after we die, we are going to run out of ATP in our muscular cells. And once we run out of ATP in our muscle cells, the myosin heads will get permanently stuck to the actin filaments, and there's no ATP that can detach them anymore. And once those actin heads get permanently stuck to the myosin filaments, rigor mortis is set in. The stiffness of death, quite literally, is what rigor mortis means. And rigor mortis can be used to determine time of death, particularly if we have good body temperature readings. So, class, concept check. When troponin is exposed to calcium ions, what happens? A, troponin changes shape, tropomyosin moves, actin and myosin come into contact with each other, contraction of the sarcomere, or E, all of the above. So go ahead and pause the video and check your text, check your notes, get me an answer. 5, 4, 3, 2, 1. The correct answer is E, all of the above. The very first thing that happens is troponin changes shape. The next thing is tropomyosin will move. And third thing will happen is that actin and myosin come in direct contact with each other. And then fourthly, we initiate the cross-bridge cycle and we have contraction of the sarcomere. So when troponin is exposed to calcium ions, A, B, C, and D happen in that order. That's all we have for this first recording. If you have any questions, please feel free to post them in the class discussion board or shoot me an email or swing by my office when you're on campus. Happy studies!